Hello, my name is Laura Murray, and I'm a second-year PhD student working with the University of Exeter Medical School and the European Centre for Environment and Human Health. I'm going to be talking a little bit today about my PhD project, which is titled Selection and Co-Selection for Antimicrobial Resistance by Plant Protection Products. Bacteria can become resistant to antibiotics, and these are the chemicals designed to kill them. This occurs under selective pressure, where bacteria are put under stress from exposure to an antibiotic. The susceptible bacteria are killed off, but those with resistance survive and continue to grow. The antibiotic is therefore ineffective. The environment is recognized as a contributor to antibiotic resistance due to a number of reasons. First, environmental compartments such as soils and rivers are home to diverse communities of bacteria, some of which may naturally harbor resistance. It's also subject to antibiotic selective pressure. This selective pressure happens as a result of antibiotics being discharged into the environment. For example, waste from production or use, residues found in livestock manure, or from the application of antibiotics to crops for disease prevention. Resistance can be maintained in the environment, and for this reason, it can be considered a reservoir of resistance. Part of my PhD is looking at the antibiotics which are applied to crops and the subsequent effects this may have on environmental bacteria and resistance. I'm considering differences between antibiotics, effects of concentration, mixtures, and repeated applications, but also differences between exposed communities, for example, if there's soil or sewage. Currently, I'm comparing a clinical antibiotic, one used on crops and one known to be used in both circumstances. This table shows usage concentrations and the lowest concentration resistance may be selected for. This was worked out from lab experiments and is lower in all cases. Using these concentrations, I'm conducting experiments to investigate community changes and gene increases. Other agents can exert selective pressure. Bacteria can develop resistance to metals, and this resistance may be effective against antibiotics or linked to antibiotic resistance. It's referred to as co-selection, as resistance to two different agents are linked. It's of great concern, as so long as the metal is present, antibiotic resistance can be maintained in the absence of the antibiotic. My PhD will look at whether non-antibiotic plant protection products have this co-selective ability. The chemicals are famous for affecting non-target organisms and are applied directly to soils containing bacterial communities. Similar to crop antibiotics, I will be investigating increases in genes and community changes. Bacteria can share resistance genes. Environmental bacteria may share resistance with human pathogens. Humans may then become exposed to the pathogen, for example, via contact with soils, bathing waters, or edible crops. This could result in resistant bacterial infections. The project is case-funded by AstraZeneca and the BBSRC, and the area of AMR in the environment is attracting strong interest from policymakers. Thank you very much for listening. Pictured are my University of Exeter supervisors, Amy, Will, and Lee Hong. Jason Snape from AstraZeneca and Alistair Boxall from the University of York also make up part of my supervisory team.